13 is going to focus on the viruses and prions. With COVID-19 going on, obviously even the general public is more aware of viruses these days. We're going to talk about general characteristics of viruses and also talk a little bit about COVID-19 itself. But before we get into that information, it's important to realize that viruses are not bacteria. People often confuse these and put them in the same category, mostly because both can cause disease. They're spread by similar routes, by respiratory droplets or other contamination from people to people, animals to people, and animate objects. And therefore, we think of it as this kind of pool of infectious diseases, and that's why people often think of bacteria and viruses almost as being the same. However, bacteria are actually cells. They are individual cells that cause disease. They are responsive to antibiotics. Viruses, on the other hand, are not cells. They're just particles. They're much smaller than a bacteria. In order for them to reproduce, they actually have to infect into a host cell, hijack their machinery to be able to replicate. They are not responsive to antibiotics. They have their own antivirals. And so even though we're often trying to distinguish whether infections are bacterial or viral, bacteria and viruses are very different. All viruses really are genetic material with a protein coat. Viruses can contain DNA or RNA, but they don't contain both at the same time. So some viruses can have double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA, but they're not going to have DNA and RNA at the same time. They have to have a protein coat, what is referred to as the capsid. It's the protective layer. In the picture here, we see the genetic material, the nucleic acid, is the purple squiggly line, and then the capsomeres, the individual protein units, are the little blue balls. Everything else to a virus is an upgrade. Some viruses, but not all, have an envelope. Some viruses, but not all, have these protein spikes that may help them to attach or release from cells. Most viruses are very specific. They only infect a certain type of cell. That is why we talk about respiratory viruses versus GI viruses. They are going to have specific attachment sites that are going to define their host range and specific cell type. So that is why, in general, we don't see that we get a cold from our dog, and we don't give our dog or cat our cold. However, we will see that things do jump species, and that is the case with both the flu and COVID-19. So in general, the host range is determined by these specific attachment sites. However, we can see some crossing over. Typically, when I talk about viruses, it was always using the flu as the model virus. Influenza virus, which is specifically the orthomyxovirus, is well tracked and documented. The CDC each year has their flu view. This is a great resource that talks about what the common strains are, how effective the current vaccines are, things like that. They're always tracking the numbers and the different types, and so we will continue to use that as a model, but I'll talk a little bit about some of the comparisons to COVID-19 as well. So right now, we have this influenza type A, H1N1, that was a holdover from the 2009 swine flu that still is the most commonly reported influenza virus this past season. So the H1N1 swine flu in 2009 was one of the, harbingers of that we were concerned about the next flu pandemic. So it didn't really turn out to be a flu, but you kind of get the same story when we think about the flu and COVID-19. We knew that the big one was going to come. When we talk about the defining features of naming these flus, that is because they have these spikes. They have an H spike, which is hemagglutinin, and this is used for attachment to the host. There are 16 different subtypes. On the picture here, they're the little blue pyramids. The neuraminidase spikes help the virus to release from the cell, and there's only 9 different subtypes. Those are the ball and stick green model on the diagram seen here. So in 2019, here's the tracking of last flu season. We want to track both the A and B types. Type A is the most serious and what is going to be causing epidemics and pandemics. Type B can still be problematic, but usually is more localized. There is also a type C that we don't track just because it's very mild. So if you look over the flu season, we see that we have both A's and B's being reported, and then we can break that down even further. In the right-hand graph, we see that not only are we looking at the A and B types, we're looking at the most common of the types for the A's, for the B's. And so that's why we can say that the kind of peachy color bars there, that is the H1N1 from the 2009 holdover that seems to be still the most problematic out there at the moment. So that's why the CDC is always tracking. First of all, people, I think, have gotten the message that the flu can be deadly. The CDC estimates that as many as 56,000 people die annually from the flu in the United States. That number varies widely according to the flu season, but it does help us to make the general public understand the need for getting their flu vaccines, and that when people say, oh, it's just the flu, that's not necessarily true. 2017-18 was a particularly bad flu season, over 61,000 deaths in the United States. 2018-2019, not quite as bad, but we still are going to see significant deaths from the flu. And now when we add that to COVID-19, people are very concerned about the coming winter. So in order to track the flu, we do this by the spikes. Again, those H spikes and N spikes are going to be very useful in naming the flu for that particular season. And this occurs because of what are referred to as antigenic shifts and drifts. Antigenic drifts are minor changes. This can be as subtle as a single mutation in one of the spikes that may even involve only one amino acid, but it changes it enough that it allows the virus to look a little bit different to your antibodies. At your mucosal membranes, you have specifically IgA as the main antibody that is there at the ready trying to bind these flu viruses so that they can't infect and cause more harm. So getting around that first line of defense of those IgA antibodies does mean that it is more likely to cause disease. Antigenic shift, though, is the real problem. These are major changes that we have in the H and N spikes, usually due to genetic recombination. We'll see that part of this crossing over species leads to this genetic recombination that then gives us new flu types that are problematic. As I said before, the viral strains from the variation in the H and N spikes are then further subdivided into these type A, type B, and type C, A being the most severe, causing the epidemics. Notable ones recently, the H1N1, H3N2, H2N2s have all been notable in some of our fairly recent outbreaks. Type B is kind of the moderate local outbreaks that we see all the time, and C is the mild disease. So when we look at the flu strains, H1, 2, and 3 are typically associated with humans. So that's why when we saw the H1N1, H1N2, H3N2, they have to be able to infect humans. The animal types are usually H4, H5, H6, and H7. All A subtypes are found in birds. And so that's why we worry about bird flus, because those avian flus start to become the next flus and possibly the next flu pandemic. So in the 1997, we had what was referred to as this avian flu from H5N1. People got really nervous about this because it had a 90 to 100% mortality rate in birds within 48 hours. That meant that this had the potential, if it crossed over, to be a really bad human flu. The H5N1 itself did have a few confirmed human cases, 190, so not a trivial number, but it never quite became the epidemic in humans that it was in the bird population. However, we see the differences between these different strains. The H1N1, which was the swine flu back in 2009, it was easily spread but rarely fatal. So a lot of people got it, but the good news is that it wasn't quite so severe. In contrast, the H5N1 spread slowly. It wasn't easily transmitted to humans, and most of the confirmed cases were from people being around the birds. The problem was, though, it was usually fatal. And so we knew that we had a bad strain, and that's what we're always trying to combat and predict. So the model for antigenic shift is that most of our flus start out as an avian bird form. The problem is that pigs are a little promiscuous in the types of viruses that they accept. They can be infected by bird flus, and they can also be infected by human flus. That makes them this unique kind of recombination spot where you end up making these new recombination of viruses that have some of those bad bird flu genes. You start to humanize it because the pig is also infected with these human flus, and you get a new virus that now humans have very little protection to. So that's why we're always getting these new flu strains, and that's the model for antigenic shift. The H5N1 that we talked about that was particularly problematic for the birds also was unusual. Not only did it infect pigs, but it was also infecting some of our cat populations. This included some of our big cats, tigers, and leopards, which are already endangered, but it also was able to infect ferrets and domestic cats, which was a little bit unusual and a bit concerning for how this could be passed on from possibly pets to humans. So the last pandemic that we had was what was the swine flu in 2009, the H1N1. This became more notable because the H1N1 was the worst flu on record back in 1918. So it was concerning that this 2009 variety had some of the genes from that 1918 H1N1. That wasn't the sole source of its genes, though, so it wasn't just that the 1918 one was back. It started with an avian gene pool and then had this triple reassortment. Some from that 1918, some from the American swine population. The human H3N2 was contributing, and you got this reassortment that gave us an H1N2 variety. However, there was a fourth component. The Eurasian swine kicked in the N1, and so then we got this 2009 H1N1 variety that caused the pandemic that fortunately wasn't quite as bad as what we anticipated, but it was basically one of the things where we started to realize we did not have enough vaccine, and the preparation of how we were making vaccines, mostly in eggs coming from birds, was problematic. So the three major types of A, B, and C are going to vary widely in how severe they are. The most severe, the type A, and that's why we see the type A pandemics. The worst on record, 1918 through 1919, that H1N1 had 50 million deaths. And so it was just warehouses of people that were sick with the H1N1. In 1957-58, we had an H2N2, 70,000 deaths specifically in the United States, about 1 million worldwide. The 1968-69 H3N2, 34,000 deaths in the U.S., and again, probably about 1 million worldwide. In 2009 and 10, that H1N1 didn't get as bad as what people thought, but it was a wake-up call that we really needed to start tracking these better and thinking about how to defend against the next pandemic. So that's why we not only have the CDC Flu View, which tracks the flu annually and gives us all sorts variety of information about the flu as pictured here, but it was also developed what is referred to as the flu site. This is for forecasting what is possibly the next flu that could be the next pandemic. So looking at the potential for impact and then trying to reduce that impact. Because we are still seeing in places like China, mostly agricultural societies, we often look to the east to predict the next bad flus. We still see a large population of people in the east, in places like China, where people are in contact with a lot of their avian farm animals and such. And so that seems to be where the flus come from. And that's why we are always trying to get a jump ahead of what's going to be the next one coming to the United States. And the CDC is always tracking and trying to better plan ahead. We also have the CDC FluVax View, which basically talks about how good the current vaccines are. Some years, the vaccine is better than others, and so it's important to know if there are certain populations that maybe just aren't being immunized well, for example, our elderly or something. So that's why each year the CDC is trying to predict what should be the next flu vaccine. The current flu vaccine for 2020-21 is going to have the base of a trivalent, and this is the one that is an egg base. We still traditionally use eggs to make a lot of our vaccines. So we have this Guangdong variety, the Hong Kong strain that has been updated, and then a B. Washington variety that is also been updated. So 2 A's and a B. However, most of the vaccine that's going to be on the market actually is going to be the quadrivalent form. So this is going to have those 3 viruses plus an additional influenza B vaccine that was around from last year, this Fuket B strain that is from the Yamagata lineage. The egg base can only be given to those people that are not allergic to eggs. So we also have other recombinant-based vaccines that are going to be similar but a little bit different. We see that the recombinant quadrivalent that is being available has instead a different Hawaiian-based strain in it. And so, again, there's little slight variations that people may not realize. Even when you go and get a flu vaccine, you may be getting a slightly different version than somebody else. For the elderly, they usually have to get a version that has an adjuvant in it. They don't respond well to vaccines with their waning immune system, so they may need an extra boost, basically, in order to help. And so that's why, again, we don't think about the different vaccines, but there are different vaccines being made by different companies. And so depending on where you got your flu shot, that is going to determine which strain specifically. But the most problematic ones are usually overlapping between them all. So that is why we're very concerned about the coming winter. We're going to have the flu season, and we're still in the midst of COVID-19. How the coronavirus and the flu are alike but very different. So yes, they have the same respiratory transmission. They overlap in signs and symptoms, which is going to be very confusing in trying to have people identify whether they think they have the flu or COVID-19. But the real issues are the ones that you know. Hospitalization rate for the flu is about 1 to 2%. COVID-19, 10 times that, approximately 20%. Length of hospitalization, when needed, about 5 to 6 days for the flu. COVID-19, on average, is about 11 days. Death rate is the main issue. For flu, even for some of the bad flu pandemics, is about 0.1%. For COVID-19, 10 times, this is about 1%. And when you look at that percentage, sometimes that allows people to minimize, but we can't allow that to happen. It is problematic. It's 10 times more deadly than the flu, which is already deadly during the winter months. That's why we're very concerned about the coming winter. Same as the flu, though, for COVID-19, it's those spikes that hold the secret to its infectivity. So that's why we're learning a lot about these spikes of the virus. COVID-19 is really SARS-CoV-2, and we see here the spikes that are on the surface there. These are going to interact with the receptor, what is referred to as ACE2, that is on human epithelial cells. ACE stands for angiotensin converting enzyme, and it's kind of this place where we're targeting most of our vaccines. So viruses are always problematic. We have antiviral drugs, but the problem is that these are going to have toxicity often to the host from the standpoint that the virus has to get into the cells to be able to replicate. We do, however, have some of our typical flu antiviral drugs. So the neuraminidase inhibitors, these inhibit the viral release. We have things like Relenza and Tamiflu. These have been effective, and it was effective for that bad H5N1. When we have some of the other drug classes, the adamantanes, these inhibit uncoating. These are useful, but as we see, the H5N1 was actually resistant. And so therefore, sometimes we don't have our whole arsenal of our antiviral drugs. It's one of the reasons that for viral diseases, we do rely on vaccination, and we'll talk about that more in Part B of this chapter. So for COVID-19, there's currently no cure or vaccine at this time. We do have some things that have been promoted. The steroids, anti-inflammatory and immunosuppressive drugs. You've heard about the remdesivir. This originally was being investigated to combat Ebola, and antibody therapy or passive immunotherapy. You're aware of these because of the triplet of drug therapy that the president recently received. The dexamethasone, the steroid, is pretty much becoming standard care for COVID-19. It is an anti-inflammatory drug, immunosuppressant, commonly given to people who have autoimmune diseases, and it works by simply suppressing the immune system. What they found is that it does cut mortality by about a third among the severely ill, especially people that were on ventilators, and by a fifth for patients receiving supplemental oxygen. There's really no benefit to people taking this steroid with mild disease, and it does have some side effects, which really could mean it could do potentially more harm than good. So it's only recommended for the moderate to severe, which is what we suspect that President Trump was on the way to a more moderate disease. Later in the course of the disease, this is when this is important. What usually has been happening is that the COVID-19 causes the immune system to be hyperactivated. You get cytokine storms and the release of all these inflammatory cytokines. This damages the lungs and other organs, and that's what typically happens to the people who become severely ill and also tend to have that precipitous drop in their health. So the dexamethasone is helping these patients by tamping down their immune response. The remdesivir is a typical antiviral drug. It's a nucleoside analog that stops the virus from spreading. So in order for the virus to replicate, it needs polymerases to take the nucleotides and putting them together in the growing chain of DNA. And so the remdesivir looks like one of the nucleotides, but doesn't allow the next nucleotide to be linked on, and so halts the replication of the virus. It does have to be given by IV infusion. He also received the REGN-COV2 by the Regeneron pharmaceutical company. They develop monoclonal antibody therapies. What this is based on is natural antibodies from patients that have survived. So you can give somebody passive immunotherapy. It means that you're giving them antibodies that are going to be able to respond to the virus. They do not last long-term. They don't cause that person to be long-term immune, but it's going to help take care of the initial stages of the virus. So they're using these natural antibodies that they have isolated and making monoclonal antibody therapies using these as the model so that hopefully we can get enough of this passive therapy available, but it's still experimental and still in limited supply. Vaccine trials are progressing. There's a lot of vaccines in the pipeline. 4 that have made it to the phase 3 clinical trials have been the ones that are probably most prominent in the news at the moment. These have all been part of what is referred to as Operation Warp Speed. This is an attempt to have a breakdown of the different companies trying to be the winner here. We need to have a multi-pronged approach, and this collaboration meant that we're trying to accelerate the development, manufacturing, and distribution of any countermeasures for COVID-19. So it's kind of like, let's support lots of winners, and hopefully we can combat. As Dr. Fauci has said, it's likely that multiple COVID vaccine regimens will be required in order to meet the global demand. Some of the ones that are in the phase 3, we see the J&J, that is by Johnson & Johnson, specifically the Janssen company. This one is using a human adenovirus in order to put the spike protein into cells. One of the reasons this one has especially been notable recently is because it may be protective after a single dose. Some of the other ones may require multiple doses. The other one that has gotten lots of attention, the Moderna, this is a one again focusing on the spike protein, but this is using a messenger RNA platform to deliver the spike immunogen. The Oxford group and AstraZeneca, they are using a non-replicating chimpanzee adenovirus, again, to deliver the spike protein, so a more traditional approach to inducing the immune response. And then this last one by Novavax. This is a Maryland-based company that actually has never taken anything to market yet. So it's kind of unusual that they're up there. But they have a protein-based vaccine where this is a perfusion protein that they're using a nanoparticle technology to deliver. The other thing is that they have this proprietary adjuvant in the mix, and that may be really important for our elderly population who aren't going to respond well to the vaccine. If you look, and we'll discuss this more in our discussion board that will be assigned this week, there are lots of other ones in the pipeline, and hopefully we come up with something in the near future. Will it be before January? That's uncertain at this point, but hopefully the start of next year, some of these vaccines may be available. So that's why in looking at the life cycle of COVID-19, those are going to target with the vaccine the attachment and entry. Don't let the virus get in. When you talk about antivirals, they are usually going to target when you already have an infection, so prevent things like uncoating or prevent the virus from making its DNA, things like that. You need both strategies. You can't rely that everybody is going to be immune by vaccination. You need to be able to treat. So we need to develop both the vaccine and other therapies to combat the current pandemic. So unfortunately, the time is right for the COVID pandemic, and we hope that we're not going to have a bad flu season. But conditions for a flu pandemic exist all the time. We are constantly seeing these new influenza subtypes being generated, and it comes when we have those antigenic shifts where people have little or no immunity. The virus that is going to make the next pandemic has to be able to cause disease in humans. That sounds like, well, of course it does. But the rationale is that most of these are starting out as some sort of an animal virus in an animal reservoir, so it needs to be able to make that crossover to where it's really infecting humans and then becomes easily spread human to human. That is what we're still trying to figure out with COVID-19. We suspect that this was from bats. Again, bats are teeming with lots of different coronaviruses, and that means you have lots of opportunity for recombination events and things to be able to generate novel viruses. The bats are very common, and they're the second most numerous mammals after rodents. They live in large bat colonies in caves, and so they become these incubators for these infections. We think that this then somehow in the wet markets in China was able to be spread to humans. There's still a lot of controversy about what may have particularly happened. You saw people talking about maybe this had gone to pangolins, which are also in those markets, and crossed over there. But we're fairly certain that the original virus was carried at one point in the bat population and now has become humanized for the human-to-human spread that we have with COVID-19. So to put into perspective, when we started with the pandemic, it was first declared a pandemic on March 11th in 2020. As of April 22nd of this year, we saw that COVID-19 already in its death toll had reached 180,000. That was simply an astronomical number in general, but in putting it in perspective is what the cute little fuzzy spiky balls are showing here, is that yes, it's nowhere near what the Black Death was. This in the Middle Ages was millions, 200 million deaths from 1347 to 1351. Yes, this was the bubonic plague that was changed world history, but it did take a couple of years for that to happen, and it was at a time where modern medicine as we know it, wasn't available. Smallpox is up there, but it's still ongoing, although we are this close to eradicating smallpox. What's a more valid comparison is the Spanish flu. Despite the name of the Spanish flu, it did originate in Asia, but it got its name from where it started to spike. So we see that the Spanish flu, the worst flu on record, was 40 to 50 million deaths. This was also, though, at a time where we did not have the treatments and vaccines that we currently have. When we try to put this in perspective, though, initially it was still looking like, oh, it's not as bad as like the Asian flu, that 1957 one, or the Hong Kong flu in 1968, Russian flu as well in the late 1800s. We see, though, that this is not the end of the story. It was ongoing then. If we see the updated numbers, as of October 11th, we have surpassed the 1 million mark. So this puts it in line with the cholera outbreaks, the Russian flu, the Hong Kong flu, the Asian flu. These were at around that 1 million, but that took a little bit longer, and we are certainly still ongoing. So that's the concern. We are trying to prevent this from going to the magnitude of the Spanish flu. Hopefully with drugs, vaccines, we will get there. But this probably would have been on platform with the Spanish flu if this had happened in something like 1918. And so we shouldn't be cavalier about the fact that, yes, we are making progress against the virus. It is still a very serious threat. The other thing is the ability for this to spread. We measure this by what is referred to as the R0. This is a basic reproduction number of disease that looks at how easily viruses and other infectious diseases can be spread. So when we talk about the flu, it has a 1.5 R0. That means if you have a person that has it, they're likely to spread this to 1.5 people, and that means that this keeps propagating. When we talk about COVID-19, It's estimated, although some studies vary, at about a 2.5. Excuse me. So certainly more infectious than the flu, which spreads fairly easily, but not nearly as infectious as something like the measles. Measles is one of the most highly infectious at a 16 R0, but we start to realize that our trivalent vaccine, our MMR vaccine for measles, mumps, and rubella, we see mumps at a 4.5, rubella, which is German measles, at a 6, and measles at 16. That meant that these are diseases that are easily spread, and so vaccination was a good strategy. We'll talk about more vaccines and the different types in Part B of the slideshow. Not everything is about the flu and about COVID-19, although it is preoccupying us for the moment. We need some general information about the viruses too. So viruses obviously are very small. The comparison here in size, the E. coli bacterium to the bottom there and the red blood cell over to the right-hand side, we see virus particles are various range and shapes. Ebola, the candy cane shaped one, is rather large compared to something like the poliovirus, which is very, very tiny. And we have just about everything in between. Viruses were first found because we could filter them away from bacteria. So if you had a filter with a pore size that was allowing bacteria to go through, you got both bacteria and viruses. And so that's why it was hard to talk about them as separate infectious agents. By using a very small filter pore size, we could then trap the bacteria, but still allow the virus to go through. So the green balls here represent the virus going through, and now we had a liquid that contained just the viruses to be able to study them. So viral structure, it's hard to get this across because they're not cells, they're just particles, but they still have physical and chemical structure that we need to talk about. So here we see a couple of pictures of what the Methadona adenovirus looks like, the herpes virus. They are defined particles that we can see mostly using an electron microscope. The typical viral structures usually are either a helical virus, where the protein capsid is going to basically be a barrel. The individual capsomeres are the little balls there, those individual protein units. They line up to make this tunnel of the capsid, and then the nucleic acid is usually very organized as a helical twist. So Ebola is an example. However, when most people draw a typical virus, they usually draw the polyhedral form. Then a 2-dimension, they'll draw it as a 6-sided figure, but it's really in 3-dimension, all these triangular surfaces, and then the DNA, the nucleic acid, is just kind of a spaghetti mix inside. We also have complex viruses that aren't just so simple as the polyhedral or our tubular forms. So these are referred to as complex viruses. The complex viruses are going to have additional things. We're going to talk about bacteriophages. Even bacteria have viruses that infect them. And we have learned about the complex viruses that are these bacteriophages about replication. The interesting thing about these bacteriophages is that they don't actually enter into the cell, they only inject their DNA. So the picture here shows the capsid head being emptied as the virus is injecting its DNA. Other human viruses, such as the orthopox virus, is also considered a complex virus, so doesn't have just the basic structure. In addition, some viruses, as we said, can have envelopes. The flu virus is one that has the envelope with those important spikes to the outside. The envelope is going to actually be a phospholipid bilayer, so it's another membrane that the virus can pick up from the host cell as it exits, and this is going to contribute to the overall morphology of those envelope viruses. We see here pictures of various ones. So we have the more tubular shaped one for the tobacco mosaic one, the typical adenovirus, the flu, a typical bacteriophage. They all look very different, but they're very small, and we see that they're going to have different strategies for their replication. Taxonomy of viruses is also complicated. Similar to what we talk about with bacteria, they have a family, a genus, viral species, but they don't follow quite the traditional naming. When we talk about the family name, it ends in viridae, the genus name ends in virus. For the viral species, this is going to be defined by viruses that share the same genetic information and ecological niche. They infect the same host. What we have started doing, though, is trying to clean up this naming. A lot of viruses were named according to what somebody thought about the way the symptoms looked or something about the virus itself. So, for example, you're familiar with the herpes virus. The family name would be Herpes viridae, and then the herpes virus is the genus. But we designate these by subspecies that are designated by numbers. This is very important. We've started referring to human herpes virus, the HHV, by then its subspecies numbers. So type 1 causes things like cold sores. Type 2, it takes 2 to tango. That's what people think of as herpes, the sexually transmitted form. But HHV3 is actually chickenpox. And so therefore, we're trying to make this more scientific naming, but the more generic names that people recognize the viruses by still exist. For HIV, the family is Retroviridae, the genus is Lentivirus, but we have named the virus itself as the human immunodeficiency virus, HIV 1, 2, the different designations. So most people recognize them by the subspecies designations rather than by their genus or family names. So we're going to talk about the endings. So papilloma viridae would be the family, papilloma virus, the HPV that we know as the causative agent of cervical cancer, those would be the appropriate scientific names. We also have issues in growing viruses. When we talk about viruses, we see that the bacteria are easy to grow on artificial media. Give them the appropriate nutrients, add in some agar to make it a solid plate or something, and we can easily grow and do the propagation. Viruses, on the other hand, have to be grown in cells. And so therefore, this is much more complicated, growing them in cell culture or in an animal system, or we typically use eggs for a lot of our animal viruses. We've learned a lot about how viruses grow and are cultured by bacteriophages, though, those viruses that infect bacteria. Bacteriophages, they get grown on a lawn of bacteria. So before the bacteria are visible, you are going to plate them out, make a nice even lawn, and then infect them with the bacteriophages. Where the virus has infected, you will then see, after you allow the bacteria to grow up, plaques. These are zones of clearing where the virus has killed the bacteria. You can then take a plug out of the agar and get the virus out of those plaques. Animal viruses are typically grown in embryonated eggs. Many different areas of the egg can be used to inject and allow it to grow. But again, when you have something like the flu, which starts out as maybe a bad bird flu, relying on eggs to make vaccines and such can be problematic. And that's why we're looking for newer technologies. We have been growing viruses in cell culture for a while, but it has just more recently been seen the value of using cell culture in making our vaccines as well. When you grow viruses, there are different types of cell lines. You can use continuous cell lines, which are basically cancer cell lines that, similar to what you know, the Henrietta Lacks story, the HeLa cells, her cells are immortal. They live on. As long as they are given nutrients, they can just continually divide. We also typically use primary cell lines. These are from tissues. You take tissue, you grind them up in a blender, treat them with some enzymes to get separate cells, and then you plate them in a petri dish with appropriate nutrient media. Cells that are mature cells, they have contact inhibition, and they will form a nice even monolayer on the surface of the petri dish. Unfortunately, these primary cell lines, they die out after a few generations. And we can use them, though, to figure out when the virus has infected because the cells become transformed. So the normal cells will have this nice single monolayer, but if they've been transformed, then you start to see that the cells start to ball up and start to make these kind of aggregates in cell culture and are no longer a monolayer. So these transformed cells have been virally infected and oftentimes will start to look like a cluster, and almost the same as when a malignancy, a normal cell, gets transformed, it then becomes a solid tumor in many other cases. We can also use diploid cell lines for growing viruses. Unfortunately, this involves human embryos, which is controversial, and these are maintained for longer than primary cell lines, about 100 generations, and have been used for things like the rabies vaccine. But we're looking at more basic cell culture as hopefully being the wave to make most of our vaccines. Here we see the normal fibroblasts spreading out on the bottom of a petri plate. As they go through cytopathic effect, you see that they start to ball up and they look sick, but that means that they're ready. They are full of virus, just ready to burst. We also can do virus identification with more modern techniques. We can use serological tests using antibodies against the virus to see when it's present. We can also do things to look at when the patient has been exposed, and so looking for antibodies in their serum. All kinds of ways to use antibodies to help us to find the viruses. Most of these serological techniques are developed when you have common viruses, since you have to have the antibodies at the ready to do the testing. We also can do molecular biology and do nucleic acid sequencing. Similar to like doing a paternity test, you can do restriction fragment length polymorphisms. You can get the fingerprint of the virus. Many times we have to utilize polymerase chain reaction to give us enough to be able to do the molecular techniques, but it allows us to track the various viral strains and give them a fingerprint, so we can even follow this from patient to patient or population to population. And these molecular techniques have become very helpful as well, although not as commonly used. So we're going to take a look at some of the more important features of bacteriophages and looking how the viruses are able to replicate. So viral multiplication, we've learned a lot from what are referred to as lytic and lysogenic bacteriophages. Lytic means they're going to rupture out. Lysogenic means they incorporate into the bacterial genome. In the multiplication of the lytic cycle, we first have to have the attachment of the phage by its tail fibers. So it's more specific than it just kind of sitting down and injecting its DNA. For the penetration, the phage actually uses lysozyme to break open the cell wall so that it can force the tail core through to be able to inject. Once inside, the DNA is now going to direct the biosynthesis, making new phage DNA and proteins as needed, put that all together in maturation, the assembly of the phage particles, and then the release using lysozyme again to burst open the cell wall. When we talk about the lytic cycle, it's very defined in after the DNA is injected, you start to see the production of the different components. It then, in the maturation process, starts with the tail and at the same time putting the DNA in the capsid head, coats the protein around the tube of that tail portion, starts to put it all together, and the last thing that gets added on are these tail fibers. Once that is done, you see the release. When a virus grows, it almost looks like a backwards of a bacterial growth curve. It's referred to as a one-step growth curve because it hides out for a while. You first have attachment. A limited number of infective phage particles are going to attach, but as they penetrate, they're no longer detectable in circulation because they're going into the cell. That's when you have the eclipse period. That biosynthesis and maturation is making the new phage particles, and these are going to be released at the burst time. When you're talking about bacteriophages, this works on the order of magnitude of minutes, and you see the phage particles being released by the lysis. And most phage individual viruses have a specific burst size. So typically they give about the same amount of virus released each time, which makes it a nice predictable model. Animal viruses look similar. It's still a one-step growth curve. It's just curvier because it's on the order of magnitude of days, not minutes, the way bacteriophages are. So we still see the attachment, then penetration, and then the eclipse period, and then the release, that exponential release of lots of the virions from the host cell. That is when you start to experience the effects of the acute infection, and then the viral load begins to peak. Hopefully your immune system or other interventions then start to lead to the decline of the number of virus particles, and that means that you're beginning to fight off the infection. So we're going to take a look specifically at the lytic and lysogenic cycle and what this means not only in understanding virus replication, but also some health implications as well. So normally when we think about a virus that infects, in this case we're looking at bacteriophage alpha that infects E. coli, we usually think of the lytic cycle. The phage attaches with its tail fibers, injects its DNA. The DNA then starts to take over and it starts to tell the bacterial cell to make lots and lots of new phage DNA and proteins so that they can be assembled into the virions. These are then going to be released from the cell, and the virus has been propagated, which is what viruses want to do. However, sometimes you can have the viral DNA incorporate into the bacterial host genome. As the phage DNA integrates, it's referred to as a prophage. The only time it is replicated is when the bacteria itself replicates. So if it keeps replicating, then that lysogenic prophage, that is going to be replicated as well. However, that prophage can come out. If the bacteria experiences a change in its environment, sometimes this can be induced by UV light or temperature or various other stressors, it may induce that the phage is going to excise and then go back into reproducing in the lytic cycle. Lysogeny, though, is interesting because there are three important results of lysogeny. First of all, lysogenic cells are immune to reinfection by the same phage. It's not immune to all other viruses, but it can't be reinfected by that same type. The other thing that is of human disease implication is what is referred to as phage conversion. Host cells may exhibit new properties. So we can have from lysogeny that the bacteria is going to get new toxic properties. So Corynebacterium diphtheriae, the causative agent of diphtheria, Streptococcus pyogenes, the causative agent of scarlet fever, Clostridium botulinum, causative agent of the deadly botulism, and Vibrio cholerae, the causative agent of cholera, they only produce the toxins that are the disease component of these organisms when the gene is present on a prophage. So they have to get these very harmful disease-causing toxins because of a prophage in their bacterial genome. So that's one reason why we can have sometimes Strep pyogenes may be around and not causing disease. It doesn't have that prophage that allows it to produce the toxin that makes it now a more problematic disease-causing organism. We also have specialized transduction, where we see bacterial DNA being packaged with viral DNA. Transduction in general can occur as the lytic process is going on. So when the phage DNA is injected, it is basically destroying the bacterial host. One of the first things that happens is that the bacterial chromosome is broken into pieces. As the packaging occurs, you may accidentally have bacterial DNA being packaged into a bacteriophage head. That can then be released out in the environment as the bacteria is lysed open, and it can then infect another bacteria. What it injects, though, is bacterial DNA. That bacterial DNA from the first host can now integrate into the bacterial host of the recipient, and it may then pick up some new properties based on this recombination. There is also what's referred to as specialized transduction. When that prophage excises, it may not do so cleanly, meaning it can take neighboring bacterial genes with it. What that means is that that bacterial gene may be packaged with the viral DNA, so you end up with a bacteriophage that has both the viral DNA and some bacterial genes. As it injects its DNA into the new host, it's giving it not only the viral DNA, but also this new gene. That new gene may then incorporate into the host genome as a prophage, and it may allow the recipient to have a new property. The example here is that the gene that allows the bacteria to utilize galactose was in the first host. That then got packaged as the gal gene with the viral DNA and injected into the recipient host that did not have the ability to use galactose for fermentation. Now, as it has received that new gene via the specialized transduction, it now becomes galactose positive because it is a recombinant cell. So that's why it's important to look at and understand model organisms, bacteriophages being one that has taught us a lot about replication. It's one of the reasons that we actually have in our freshman sequence, our honors students participate in what's referred to as C-phages, where they look at bacteriophages that are isolated from the soil. And even though that sounds like, oh, that's an interesting project, it actually is part of a larger consortium that started out of the University of Pittsburgh, that is HHMI, Howard Hughes Medical Institute, supported. The HHMI initiative actually isolated a bacteriophage against a mycobacterium. There was a young girl in the UK that had a lung transplant and unfortunately got a incurable mycobacterium infection, Mycobacterium abscessus, that was multi-drug resistant. She was going to die unless they could do something because there was no drug therapy for her. So they allowed this experimental procedure with the bacteriophage that had been isolated through the C-phages consortium. And this young girl is still alive today. This was done back in May of 2019, maintaining on this phage therapy, which would have been a certain death without it. So understanding these bacteriophages, not only from their standpoint of replication, but potential therapy is very exciting as well. So now we're going to switch, though, to talking about the animal viruses and their replication. Animal viruses are going to be different depending on if it is a DNA or RNA containing animal virus. When we look at the multiplication of animal viruses, the stages look similar to what we saw with the bacteriophages, but one notable exception. They have to attach. They're going to attach to the cell membrane of the host cell, and we'll see how that occurs in a moment. Penetration is going to be by endocytosis or fusion, where they're actually taken into the cell. So that's very different than what happens for bacteriophages, where they just inject. So uncoating is going to be unique. The virus has been taken in, and therefore that capsid, that protein coat, has to be removed to allow the genetic material to do its job. This can be accomplished by either viral, or sometimes it uses host enzymes to take off that protein coat. Then, as we saw with the bacteriophages, biosynthesis, making all the nucleic acid and proteins as needed, maturation, assembling them all together, and then release. The release for animal viruses can either be by budding by envelope viruses, or some viruses just simply rupture out of the cell. Here we see examples. The entry of the toga virus in the top panel here is receptor mediated. So it's using these spikes and specific receptors on the host cell to be taken in, as you can see, by endocytosis. Others that have entry, down here we see a herpes virus by fusion. The membrane, or that envelope, is going to be the same as a membrane. Specifically, it's the same as the plasma membrane that it got from host cells. So therefore, sometimes it can use the fusion of these membranes to be able to, again, gain entry. And here we see, again, the fusion of the viral envelope and plasma membrane and the endocytosis, or taking in of the virus. When viruses are released, if it is an envelope virus, as it exits, it literally is budding out and taking part of the cell membrane with it. If it does that just occasionally, if it's a low viral load, that doesn't necessarily have to kill the host cell, so it can tolerate some of this virus being budded out. We see pictures here of the alpha virus budding out of a cell. But if it starts to get a very high viral load, then obviously that is going to be detrimental and kill the cell, because it can't just keep losing lots and lots of its cell membrane. Viruses that are not enveloped usually just rupture out, and that does tend to kill the cell. Here we see that budding process for a lentivirus. So you see that it's literally just using the cell membrane and using that now as the virus is released to have an envelope. Comparison of bacteriophage and animal viral replication. As we said, attachment, this is more specific than what it might seem. Tail fibers are interacting with cell wall proteins. For animal viruses, the attachment are using various plasma membrane proteins and glycoproteins. For bacteriophage, it is only injected. The viral DNA is injected as the bacteriophage squats down and lysozyme penetrates the tube portion through the cell wall to allow it to inject. For animal virus, the capsid itself and the whole virus is taken in either by receptor-mediated endocytosis or fusion. For bacteriophages, no uncoating is needed. It had injected its DNA. For animal viruses, enzymatic removal of that capsid protein is essential in order to go to the next step. When we talk about the replication, for bacteriophages, they don't have a nucleus, so everything occurs in the cytoplasm. For animal viruses, though, it depends on the type of virus. If it's a DNA virus, it has to replicate in the nucleus. If it's an RNA virus, it replicates in the cytoplasm. Lysogeny is unique to bacteriophages, but there's analogous chronic infection that occurs where we can have proviruses that integrate into animal viruses as well. This is going to happen when you have what are referred to as latent viruses. Some of these are ones that go in and out of remission. The difference is that proviruses, once they incorporate into the host genome, they never come back out, which is why we can't be cured of some of these viral infections. It also is problematic with cancers, as we'll see in Part B of the slideshow. And there's also these neurological, always fatal, slow viral infections. So these have a stage that's analogous to lysogeny, but a little bit different. But the most important thing is the fact that that provirus never is able to excise the way that a prophage is able to. So release for bacteriophages, they simply lyse out. For animal viruses, it can be the envelope viruses bud out, but non-envelope viruses, they do rupture the cell and that plasma membrane to be able to leave. We're going to talk more about DNA containing and RNA containing animal viruses. So that will be the focus of the next chapter or the next slide set. So we'll look at the animal viruses when they come in and after they're uncoded. Obviously, they release their viral DNA and the capsid proteins, but they have to then get into the nucleus. The portion of the viral DNA then needs to be transcribed in the nucleus to be able to start making the early viral proteins. This then continues with the biosynthesis and then late translation of making the capsid proteins and everything to go through maturation and make mature virions to be released from the cell. So that is going to be part A of chapter 13. So looking at the stages, especially understanding this in the context of the bacteriophage is important. And now we'll take a look in part B about RNA, DNA containing viruses and how they replicate. We'll also talk about the linkage to cancer, look at why viruses can be virulent. But we'll finish off that chapter talking about the antiviral drugs and vaccines. Viruses are harder to combat because they're in the host cell. That is why whether we're talking about the flu or whether we're talking about COVID-19, specific antiviral strategies and vaccinations are very important and are going to be how we combat some of these severe viral diseases. So we'll pick it up there in part B.